Around week 6 of intrauterine life, the physical architecture of a human mouth begins to assemble inside the primitive oral cavity. In this microscopic cross-section of the developing jaw, we see the topmost structural layer, the primitive oral epithelium, derived from embryonic ectoderm. Directly beneath sits the dental mesenchyme, distinct from standard embryonic mesoderm. Watch neural crust cells migrating from the midbrain into this exact region. Because these migrating cranial neural crust cells settle into the first branchial arch to form this lower tissue, it is specifically referred to as ectomesenchyme. The entire process of odontogenesis, or tooth development, relies on reciprocal signaling between these two distinct layers. The oral epithelium and the underlying ectomesenchyme continuously instruct one another in a precise sequential feedback loop. Neither layer contains the complete blueprint. The epithelium alone cannot determine tooth morphology, and the ectomesenchyme alone cannot trigger matrix formation. They must negotiate the precise location, shape, and cellular specialization of every individual tooth together. The primitive oral epithelium thickens, forming the primary epithelial band across the jaw. This band forks into two distinct paths, an outer vestibular lamina and an inner dental lamina. The outer vestibular lamina rapidly hollows its center, while the inner dental lamina remains solid. That hollowing of the vestibular lamina physically carves out the vestibule of the mouth, the literal empty space between your teeth, lips, and cheeks. Meanwhile, the remaining solid dental lamina begins exchanging chemical instructions with the ectomesenchyme below. Signal proteins like FGS, BMPs, and ectodisplacin cross the boundary. Chemical signaling triggers localized cell proliferation. The dental lamina pushes downward, creating enlarged projections called dental placodes. As the placode invaginates, it enters the bud stage, organizing into two cell types, an outer boundary of low columnar cells and an inner core of polygonal cells. Directly beneath this advancing epithelial bud, the responding ectomesenchymal cells begin to densely cluster or condense. The formation of this bud, with its accompanying mesenchymal condensation, is the first localized structural commitment to building a specific tooth. The transition to the next morphological stage relies on a mechanical property called differential proliferation. This heat map overlay tracks the decision rate of the cells. The cells on the outer periphery of the bud are dividing rapidly, while those in the center divide much slower. Growing faster at the edges, the structure buckles, folding the bud into a cap. This cap establishes three epithelial layers: an inner enamel epithelium lining the depression, an outer enamel epithelium on the exterior, and a central mass called the stellate reticulum. The stellate reticulum alters the physical mechanics of the cap. These central cells begin secreting complex sugar molecules, glycosaminoglycans, directly into their extracellular space. At the cellular level here, we can see how those glycosaminoglycans act. They rapidly draw water molecules from the surrounding tissue into the center of the cap. The influx of water creates immense osmotic pressure, physically forcing the cell membranes apart. The cells stretch into star-like shapes, maintaining their grip on one another only through thin intercellular links called desmosomes. While the center swells, a specific cluster of non-dividing cells remains anchored near the inner enamel epithelium. This is the enamel knot. The enamel knot functions as the primary signaling center for the tooth. Its location and the density of its signals dictate exactly where and how many cusps the final tooth will have. The macroscopic shape of the developing tooth is not drawn from a static blueprint. It is entirely driven by the physical mechanics of localized osmotic pressure and targeted signaling hubs forcing the tissue to fold. As the inner layers continue to proliferate against this pressure, the cap invaginates further, deepening into a bell-shaped structure. Watching the cap elongate into a bell shape, notice how the water-filled stellate reticulum eventually collapses, physically bringing the outer and inner enamel epithelia closer together. During this compression, a new layer of spindle-shaped cells forms between the stellate reticulum and the inner layer, known as the stratum intermedium. This late bell stage is the site of morphodifferentiation. The physical folding is complete, and the final shape of the crown, whether it will be an incisor, canine, premolar, or molar, is permanently established. Simultaneously, the tissue undergoes histodifferentiation, converting these generalized embryonic cells into highly specialized matrix-producing factories. Tracking the epithelial lineage at this boundary, the inner enamel epithelial cells convert into enamelblasts. Their sole function will be to secrete enamel matrix upward. On the other side of the boundary, the condensed mesenchymal cells of the dental papilla convert into odontoblasts. They will secrete dentin matrix downward, in the opposite direction. As the enamel and dentin matrices mineralize, the original dental lamina tethering the tooth to the surface disintegrates, separating the entire tooth germ from the oral epithelium. If this disintegration is incomplete, remnants of the tissue known as epithelial pearls are left behind. Clinically, these isolated cell nests can trigger anomalies like supernumerary teeth or odontoas. The Bell stage marks the definitive pivot in tooth development. The exact macroscopic geography of the crown and the microscopic cellular factories required to build it are finalized simultaneously. Building the root is a secondary cascade that only begins once the crown's development is heavily underway. Returning to the full Bell structure, look at the bottommost edge where the inner and outer enamel epithelium meet. This is the cervical loop. The cells of the cervical loop proliferate strictly downward, forming a two-layered structure called Hertwig's epithelial root sheath, or HERS. HERS acts as a downward guide track. Its limited growth potential strictly dictates the final length and shape of the tooth's root. As it migrates downward, HERS chemically induces the adjacent mesenchymal cells inside the ring to differentiate into root odontoblasts, which immediately lay down root dentin. Once the root dentin is formed, the epithelial tube of HERS must undergo a mandatory structural failure. It breaks apart, disintegrating into a fragmented network called the epithelial rests of Malassez. This programmed destruction is critical. By fragmenting the epithelial barrier, cells from the surrounding mesenchymal sac, the dental follicle, can finally migrate inward and make direct contact with the bare root dentin. Contact with the dentin triggers the dental follicle cells to differentiate into cementoblasts, which begin depositing a protective layer of cementum over the root. Other follicle cells in the surrounding space differentiate into fibroblasts. They synthesize the periodontal ligament and the Sharpie's fibers required to anchor the tooth in place. Finally, the remaining outer follicle cells differentiate into osteoblasts, constructing the surrounding alveolar bone that the ligaments will embed into. The structural anchoring of the entire tooth requires the intentional destruction of the epithelial root sheath, clearing the path for the surrounding mesenchyme to execute its final specialized tasks. Shifting the timeline backward from root formation, we find that permanent teeth begin their own development early on, around week 14 of intrauterine life. In this cross-section of an existing deciduous tooth bud, notice the initiation mechanism. A tiny extension buds off just behind the original structure, on the lingual side. This secondary offshoot is the successional lamina. It gives rise to the permanent teeth, pushing through the exact same sequence of localized cellular proliferation we just mapped: the bud stage, the cap stage, and the Bell stage. There is one mechanical exception. The permanent molars do not have deciduous predecessors to branch from. Instead, they bud directly from the posterior dental lamina starting at week 20, continuing their development for up to 5 years after birth. Permanent dentition is not a separate biological process. It is a targeted, secondary activation of the exact same epithelial mesenchymal pathways established at week 6. This split-screen comparison maps the week 6 embryonic baseline directly against a fully mature anchored tooth. The initial ectoderm-derived oral epithelium manufactures only the enamel. That top layer is solely responsible for creating the hard outer cap of the crown. Every other specialized tissue in the structure, the dentin, the pulp, the cementum, the periodontal ligament, and the alveolar bone, is exclusively a product of the underlying neural crest-derived ectomesenchyme. The functional human tooth is the direct result of a continuous dialogue between two cell types, executing millions of localized, chemically and osmotically driven decisions with flawless mechanical precision.